The world of DNA.
Welcome to the welcome segment of our video. Today, we're going to be talking about protein synthesis and DNA replication. Let me just get my DNA mark. Enzymes that you'll need to know for each process. First of all, DNA helicase unzips the strands of DNA. Primase creates RNA primers. DNA polymerase creates new DNA, and there are two types of DNA polymerase. Exonuclease removes the RNA primer. Ligase binds together fragments of DNA, and single-stranded binding proteins hold the DNA in place throughout the process. Lastly, spliceosomes remove RNA introns.
Welcome to DNA replication. Fasten your seat belts, kids. So we start out with some regular DNA and just the nucleus of some random cell, and then the amazing, complicated process of DNA replication ensues. When the DNA is ready to replicate, an enzyme called DNA helicase comes and unzips the strand. And you know, if I could be an enzyme, I would be DNA helicase, so I could unzip your genes. Secondly, the enzyme primase adds on a RNA primer to each separate strand of the original DNA. The next step begins with DNA polymerase 3. Begin building new DNA by matching new nitrogen bases of the original DNA. The DNA polymerase 3 can form a continuous strand of new DNA on the leading strand. However, because new DNA can only be built from the 3' end of the original DNA, the lagging strand of DNA must be built in the opposite direction of the leading strand. The lagging strand must be built in the segments called Okazaki fragments. Okazaki fragments are short DNA fragments formed on the lagging strand during the process of DNA replication. After that, DNA polymerase 3 continues to build DNA on the leading strand. Exonuclease removes RNA primer. DNA polymerase 1 builds DNA in areas where exonuclease has removed the RNA primer. DNA polymerase 1 replaces the RNA primers on both strands with new DNA. Meanwhile, primase adds another RNA primer to the lagging strand in preparation for the formation of the next Okazaki fragment. What did one Okazaki fragment say to the other? You're always lagging behind. Sixthly, DNA polymerase 3 continues to form new DNA on both strands. DNA polymerase 1 replaces the RNA primer on the lagging strand with new DNA. Ligase binds the new DNA formed by the DNA polymerase 1 to the rest of the new DNA on the leading strand. The other day, I was talking to my friend Ligase, and I asked, "What you been up to?" Ligase replied, "Oh, you know, just making ends meet." Ligase binds together the Okazaki fragments of the lagging strand containing one continuous strand of new DNA. If I were an island full of DNA, I would be
Stranded. Where's my laugh track?
You're not funny!
Anyways, we're going to start on the portion that deals with protein synthesis now. First, DNA helicase will unzip the DNA. Secondly, RNA polymerase begins to assemble the mRNA by matching the mRNA's nitrogen bases with the corresponding DNA bases. Adenine will now pair with a new enzyme called uracil instead of thymine. Cytosine and guanine will still pair together. After the complete strand of mRNA is made, spliceosomes remove the introns, which is just extra junk, and will bind the exons together and create a continuous strand of mRNA with only the necessary information. Then a strand of mRNA with the introns removed and the exons bound together. mRNA leaves the nucleus of the cell and goes to a ribosome. After that, the ribosome anticodons of tRNA pair up with codons of mRNA. Each anticodon has an amino acid bound to it. As more anticodons attach, the amino acids bind together, forming a chain. Last, the amino acids break away from the tRNA. Together, all of the amino acids form a complete protein.
Now that you're an expert on DNA replication and protein synthesis, go into the world and spread your knowledge. Make sure to tune in next week to learn about the 52-step process of beluga whale reproduction.